Close your eyes and picture a flowering plant. When we think of flowers, we usually picture roses, sunflowers, or other garden plants with colorful petals. But the flowers of angiosperm comes in all shapes and size, along with their other parts like stem, leaves, fruits, etc. The flowering plant's morphology includes the root, stem, leaves, flowers and fruits, regardless of which plant it is. If you look closely at a plant, you will notice that they always have roots and shoot system. And within those, the plant may or may not have flowers, seeds and fruits. Let us individually learn about seeds and flowers of a flowering plant. The word fruit conjures images of colorful, juicy and tasty platters full of fruits. Let us learn about fruits from the perspective of biology. In majority of angiosperm, when a flower is pollinated, the pollen joins with an egg to produce seed. The seed develops within the ovary, which is part of the pistil. The expanded and ripened ovary is referred to as fruit. Commonly, the enlarged ovary becomes the edible portion of the fruit. The fruit is the characteristic feature of flowering plants, which is a ripened or mature ovary. Do you think it is possible to develop fruits without fertilization? Yes. Fruits like pineapple can develop without fertilization, and this type of fruit is called parthenocarpic fruit. Bananas, figs, grapes and breadfruits are examples of parthenocarpy. All of these are similar to the normally produced fruits, but they are seedless. Now let's discover how fruits is developed. After fertilization or development without fertilization, many changes occur in a flower. The anthers and stigma withers, the petals drop off, and the sepals may be shed off or undergo modification. The ovary enlarges and the ovules develop into seeds, each containing an embryo plant. The principal purpose of the fruit is the protection and dissemination of the seed. Now let's discuss the morphology of a fruit. It has two main parts, seeds and pericarp. Seeds are fertilized and ripened ovules, and the pericarp develop from the walls of the ovary. Depending upon its nature, the fruit is dry or succulent. The structure of the pericarp varies greatly. In fleshy fruit like plum, the fruit wall is differentiated into three layers: exocarp, the outer layer; mesocarp, the fleshy middle layer; and the inner endocarp. We have been talking about fruits, but haven't mentioned what a seed is. How is it formed and becomes a mature plant? Let's see. A seed is a basic part of a plant which is found enclosed within the fruit. It is made up of a seed coat and an embryo. A seed is defined as the ripened or fertilized ovule which contains an active embryo. It is the reserve food protective covering. Here you can see that the seed is covered by the seed coat, and inside it a young miniature plant is present called the embryo. The function of the seed coat is to protect the embryo from injury and drying out. You can also see another significant part of the embryo called the cotyledon. It is the embryonic leaf in seed-bearing plants. The angiospermic seed may have either one or two cotyledons, that is, monocotyledons and dicotyledons respectively. First of all, dicots and monocot seeds are different. Recall that a dicot has two seed leaves in the plant, while a monocot has one seed leaf. Let us explore the structure of dicot and monocot seeds individually. The name dicotyledons refers to the seed having two embryonic cotyledons. There are around 2 lakh species of dicotyledons discovered to date. The dicotyledon seed is covered and protected by seed coat. The seed coat has outer testa and inner tegmen layers. On the surface of the seed, a scar-like structure is present called the hilum, where the seed is attached to the fruit. Above the hilum, a small pore is present known as micropyle. The embryo is present within the seed and it consists of an embryonal axis and 2 cotyledons. As you can see in the image, the embryonal axis has 2 ends. One end is the plumule formed as the shoot tip and the other forms the root tip, that is radicle. The cotyledons of dicots are fleshy and contains reserve food materials. Examples of dicots are cashew, almond, peas, etc. Now, do you know some seeds like castor, wheat and maize restore some endosperms during embryonic development. This kind of seed is known as endospermic or albuminous seeds. But in beans, peas and grams, the endosperm is completely consumed by developing seeds. Such seeds are known as non-endospermic or ex-albuminous seeds. Now let's move on to the structure of monocot seeds. As the name suggests, the number of cotyledon in a monocot is 1, and it is a large and shield-shaped structure known as the scutellum. The seeds are generally endospermic, but in orchids they are non-endospermic. In cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. The endosperm is bulky and it stores food. The outer covering of the endosperm separates the embryo by a protein layer called the aileron layer. As you can see in the image, the embryo is a small structure situated in the groove of the endosperm. Here you can see that the plumule and the radicle are enclosed in a sheet called coleoptile and coleorhiza respectively. Examples of monocotyledonous seeds are rice, wheat, corn, etc. We have explored the previous part of a plant, the fruit and seeds. For instance, you get accustomed to what is meant by epicarp, endocarp, mesocarp, true and false fruit, fruits classification, etc. On the other hand, we also covered seed, the structure of a dicot and a monocot seed. Are all the flowering plants belonging to the same family? Any guesses? Let's explore the concepts of different families of angiosperm in the next session. Now let's summarize what we have learned.
Manorama Horizon. Learning made simple.